Thank you for coming at our meetup. This is the first meetup that we organized in digital imaging. This is the third meetup that we do in total in our new group. And we have three speakers which are very interesting tonight, and I will present in a moment. I'm here also to tell a few words on behalf of RSIP Vision. Better? Yes. Okay. I'll say a couple of words about RSIP Vision. First, I will ask why I have two heads, and I will tell you in a few moments. But the first head is about RSIP Vision. We are an outsourcing company who makes software, custom software, to power your devices. The idea is that if a company has some trouble to recruit engineers to make algorithmic work in computer vision or medical imaging, well, we have that team, and we can outsource the project with us. The team is made of about 50 engineers, all experts in deep learning and artificial intelligence. They can help to bring new features and advanced features to your devices, and of course, we accompany the project from beginning to the end, until the ending. I don't have to tell you, since you are all here to hear about medical imaging, how important it is, and how some of this work can save lives and give a lot of satisfaction to the engineer who works on them. I have here one example, which is an algorithm that calculates the volume of the left ventricle. And I have here many more examples for chest segmentation. There is practically everything from bones to vessel segmentation, lobe segmentation, airway segmentation, and of course tumors and nodule segmentation. We do a lot of research at RSIP Vision. For instance, this example is for a work that we have done together with the Tel Aviv University for a client of us, which is Biosense Webster, a company of the Johnson & Johnson Group. And here is another research that we did, and we have presented it with Stats Medical at a major conference. It was, of course, another work with the convolutional neural network in the macroscopy area. Okay, now we have to explain the second head. Our company is very successful, and we are very happy about it, which means that we can free some of our time to do some work for the community. And what we do, we have created 3 years ago a magazine called Computer Vision News, in which we talk about projects, reviews, papers, challenges, and applications and solutions and people inside our community. This is a really community magazine that we do every month, and together with it, we have been asked by the major conferences, CVPR, the Computer Vision and Pattern Recognition Conference, ICCV, the International Conference for Computer Vision, and Meetup, which is the Medical Imaging Conference, to make their daily magazine during the conference, which means that we go there, and every day we publish a magazine saying what was the highlights of the previous day and what will be the highlights of the next day. I just want to ask a question. How many people here had never heard about these magazines before? Never heard. Okay, well, let's do the contrary now. How many people here have already had scanned at least once these magazines? Okay, so it's slightly more. Well, it's very interesting. I will give you just a few numbers about it. In 3 years, we have already published about 70 magazines, 70, 75, between the dailies and the monthly, for a total of which this summer will go over the 3 million pages in total. These are 3 million pages of scientific knowledge shared for free with the community. This is something. Is it online? It is online. It is online. And you just go on our website, there is written on the top magazine, and you enter there, and you see all the archive with more than 2,500 pages of written scientific text, which is not only ours, it's the community knowledge, and we just share it with you. That's pretty much about my first hat and my second hat. So if you one day need outsourcing work for your projects, while thinking about RSIP division, or if somebody asks you about, you can certainly tell them that you can work with us. I'm very, very honored now to introduce our first speaker, Dr. Aïcha BenTaieb, who is a research scientist at Roche. I met Aïcha for the first time 3 years ago at BenTaieb. Every year that I've seen her present at BenTaieb, people were very much impressed, so I'm very much honored to have her today. And that's pretty much it. Please, Dr. BenTaieb.
Can you hear me now?
Yes.
Cool. Okay. So yeah, my name is Aïcha. I'm a research scientist at Roche. I work on analyzing digital pathology images, which are images from tissue biopsies, so from tissues. And the works I'll be presenting today will highlight kind of the main research that I've done at SFU, so prior to joining Roche. We have like quite a there will be a lot of details, but at the same time I can skip, so you can choose whenever you want me to skip or whatever you want me to, continue talking about. So how many people are familiar with this pathology image analysis in general? Like, is there anyone from pathology? Cool. Hi. Cool. So for the others, histopathology image analysis in general refers to what's called the microscopic analysis of tissue biopsies. And the main goal of that is that you'd like to search for abnormalities within tissues. So what happens in practice is that when there is a tumor in some organ, or in lymph nodes in the human body, surgeons go in and take a sample of that tumor using resections or biopsy instruments or however form that is. And then what happens is that in order to assess whether the tumor is malignant or benign or any type of staging and so on, then you have to have a pathologist actually look at a sample of that tissue, a very thin section of that tissue under a microscope, and try to infer the diagnosis. So this is so far the only way to have actual, prognosis at that level. So what is shown on this image here is the process of going from the actual tissue, scanning it, collecting it in these microcassettes of physical tissue slides, and then looking at it under a microscope, but further digitizing what you see under the microscope and having that on a computer screen. So I should say that digital pathology in itself is not a widespread technique yet. Not all pathology labs will actually digitize their images. So it's quite a new field where in the past 10 years we've seen more and more labs equipped with these microscopes that have scanners on top that can actually digitize their images. But the common practice is to broadly look at the image and infer from what you see on the microscope, infer diagnosis. So there is a lot of room for working on these images, and like in everything in medical images, there is a lot of room for improvement of the actual clinical diagnostic process, and that's where our work comes into play. We try to come up with, imaging automated systems that would help the pathologists during their workflow. That's very good. That's a very relevant question, actually, because you may, so there's been a lot of progress in scanning these images, right? So at early stages, you may have these images that are not really reflective of the reality of the tissue. And some pathologists will still say that looking at the actual slide under the microscope is not the same as looking at it digitized. Yes. Yes. So ideally, when you scan these images, the scanner will come with guidelines on how, what's the thickness, the minimal thickness it should take for the slides that you're trying to digitize. And so broadly, it's generally around 4 micron. You can't go thicker than that because otherwise the image doesn't look the same at all. So it may necessitate them rescanning re-preparing the slide and rescanning it again to get the proper image. But there's more standardization now at that level where the protocols are, kind of generalized across the scanners that come with these microscopes. So to give you an idea how the image looks like after you digitize it, this is one sample of an ovarian cancer image where this image itself is a very high, it's a very high magnification image in the sense that you can see it at multiple magnifications, like you would in a microscope. So you have the 4x level that corresponds to the 4x on a microscope, where you have broadly an idea of what the tissue looks like. But then when you zoom in, you can get higher and higher details about the structural elements in the tissue, such as cells and nuclei and whatever forms the tissue itself. And because of that, because this is a high multi-magnification image, it is actually a giant image in terms of pixels. These are millions and millions of gigapixels that you're dealing with. So it's kind of along the big data problems and so on that you're going to have to handle. And further, when pathologists looked at these images, the abnormality within them is actually going to be only focused in a single area. So most of the image itself is healthy tissue. And then they would look in at this, and they have this amazing capacity of identifying the small abnormal areas within this whole thing. So you have this very challenging data set in which only a small portion of pixels is actually relevant for the task you're trying to do, and most of it is actually only healthy tissue. And there's a lot of exploration to do here in actually defining how abnormality can be detected, and the interplay between, the junction between abnormal and normal tissue, how that can correlate with outcome, and those type of questions enter into play. So when it comes to automating the analysis of these images, you're not just handling the fact that they are huge. You're also handling the fact that when they are acquired, there are a lot of artifacts that can come into play. So for instance, the whole process of going from the biopsy to the digitized image involves humans and operators staining the image, fixing the tissue, preparing it in these cassettes, looking at it under the microscope, and then there are, between those stages, even if you leave the slide and the tissue multiple hours outside, the tissue itself deforms, right? So a lot of things can change during the process of acquiring the images. And that includes all these artifacts that you can see here as an example, where you see inconsistent staining areas because the operator must have put too much staining in that area, or the tissue captured it in a weird way. You see contrasting effects because of the lighting in the room. You see deformed, just actually from the moment where they take the biopsy out of the organ, you see the suction effect from the tissue deforms, the cells within it. So all of these things account for all that variability that is not only between humans, but also in the preparation process. So it's a very challenging task to actually look at it by a human, so you can think of it as even more challenging for an automatic system. So in the research that I've been doing so far, we've been looking at ways to not only facilitate the analysis of these slides and speed it up for pathologists, but we've been also looking at ways to explore what can be a relevant biomarker to look at when you're trying to predict cancer. And so all of this enters in the scope of automating cancer diagnosis, which is a big term because fully automatic cancer diagnosis is not feasible. But at least automating parts of it that can be more reproducible and can lead to more reliable outcomes, then that's kind of something we can aim for. So I won't be talking about all of this, but just to give you a broad idea of what we did, we looked at multiple data sets. Some of them we've released, some of them come from already publicly available systems out there, but we looked at mainly ways to leverage deep learning and machine learning on these giant cohorts of patients, which giant applies to, really here applies to medical imaging world. It does not apply to computer vision world, where giant doesn't have the same term, right? It doesn't mean the same thing. These are not millions of data sets, but there are hundreds and hundreds of patients. So that's kind of a big data set in the sense of a medical imaging community. So the main thing that is the common pattern across the works that we've looked at is that we found that most of the time, including domain knowledge and priors that are related to the actual task we have at hand, works the best. And so that's what I would kind of be describing over two of these works, where first I'll describe how we designed a system that to automate the segmentation and the analysis of images of colon cancer in order to identify glands and predict whether an image is malignant or benign. And then I'll explain a more general framework where we looked at leveraging weakly supervised deep learning approaches to identify within whole slide images where the abnormality is, and specifically here where metastatic lymph nodes are within the tissue slide. So I'll start by explaining this first project that relates to glands as biomarkers. So it's common known when looking at tissues and analyzing them that glands that you can see in colon, but also in prostate and many other things, are very good biomarkers for the type of cancer that you have. So the shape of these glands generally reflects how malignant the tissue is. For instance, here you can see two images of a benign and malignant colon tissue, and the corresponding masks of, or segmentation masks of the glands within these images. And you can see that the shapes here are quite irregular on the bottom image, and that generally reflects malignancy for a doctor, at least for a pathologist. So what we try to do here is to have a more reproducible way to automate that process of identifying these glands and their shape and evaluating whether they relate to malignancy or not. So if you think of it, this is a typical pipeline in any medical imaging process where you are driven kind of by segmentation to compute some features that hopefully will correlate with the classification that you're trying to do. So here, for instance, you would segment the glands, then you would compute some statistical features out of it, and then you would use that to classify whether the image is malignant or benign. That's a common pipeline. So what we wondered here is actually whether the other way around was actually useful or not. So we wondered whether not only classification can benefit from segmentation, but also whether knowing the class would help you better segment. So we looked at kind of this symbiotic relationship between these two tasks. And we decided to formulate this using just an autoencoder type of network, where we have a classification network, if we call it that way, which is the encoding path, and a segmentation network that is your decoding path. And then we introduced kind of mid-level layers here that we called class-specific shape drivers that actually reflect how much each pixel in that feature mask would be correlated or not correlated, what is the ranking of each of those pixels to the final class label. So that way it kind of gives you this heat map of where each pixel, the importance of each pixel to the final class. And you could use them, all these feature maps aggregated, you could use them to augment the input to our segmentation path, and then use that to finally segment the whole image and move over this. So because we want to do both tasks, classify and segment, the total loss function that we used to compute this was actually the task loss of a classification cross entropy, as well as a pixel-wise cross entropy term for the segmentation part. I can take applause here because I think I went a little bit fast, but if you're interested in the details of this, I can. Sure, you can ask me questions then.
Is this a way of
It's a way of integrating both tasks of classifying and segmenting. So we have this strong assumption that knowing the class label for an image and knowing that which biomarkers you're trying to segment, both of these information should help each other out for you to get a final class.
So this is different than pattern recognition.
It's still a pattern recognition problem per se, in the sense that we're recognizing these patterns and we're trying to leverage those, the output of that recognition to classify the image. Exactly. It's a more abstract. Yeah, correct. Yeah. So the objective function is at the bottom there, L total. It's simply composed of a weighted sum of a classification percentage term and a segmentation term. So both of these are simple pixel-wise percentage versus image-based percentage, right? Yeah. In the middle there, we introduced in the green boxes are what we call these class-specific shape priors. And the way we computed those is by taking kind of looking at the ranking of each pixel within each feature map, its ranking to the final class. So how much this pixel contributes to, say, class malignant versus class benign. And when you look at these feature maps, they will kind of show you the pattern of where claims are, most likely.
The system has a corpus of knowledge that is also very likely confirm errors that you're looking for, and that's why you use
yeah. So you have a training data of annotated images and segmented images, and then you're using that to train the system. Yeah. So it's that loss function is basically penalizing the system for making mistakes when looking at the ground truth or looking at the training data that you have.
Character, or you are acquiring the training data from other sources worldwide, so that you have a larger sample of the training.
So the data set itself here came from a challenge that was released at PKI, and I believe the way they acquired the images was from actually multiple centers. That's to be confirmed, but I believe it was obtained from different pathology centers, and there was one annotator, kind of labeling each image.
Is the lambda something that is a hyperparameter?
Not in this case, but I'll touch on that in a couple of slides. Yeah.
So on the x-axis, is this like the base code? The y is like the redevelopment. What about x and y?
The x and y in where? Oh, sorry, x is the input. Is that what you're asking? Sorry, x is here on the horizontal axis. So these are the successive layers. Okay, I kind of assumed that everybody knew that, sorry. So this is an autoencoder, a convolutional autoencoder. Each block represents a convolution operation followed by some activation and followed by some batch normalization. And then we are stacking those. So each box is stacked, and what you see in blue are max-pooling layers, so subsampling layers. And on the upsampling path, that's the reverse. So what you're seeing is deconvolutional operations, so the reverse, and upsampling patterns instead of downsampling, so that you get back to the original image size. Yeah.
So you have a phenotype classification of output point.
Yes. Yes. It's shown at that P of y given x. Y is your label basically for the batch, for the image.
So the F is the class.
The F is the aggregation of the green boxes that we're calling features here, class-specific special features. And we're combining them and feeding them as input to that layer. Yeah. Malignant versus benign. All of them are adenocarcinomas. It's just sometimes it's an adenocarcinoma that is malignant, sometimes it's benign. Submax. Yes, but for each one of them. No, this was submax. Yeah.
Just can you explain the middle part and how that other?
The green boxes? Yeah, so to get those, we used a gradient-based approach. So we looked at, when you're doing backpropagation to kind of train your network, you're looking at the partial derivative, right, for each, with respect to each feature map. So we looked at that, and that's how we kind of ranking each pixel based on its contribution to the final loss function.
So the ranking, how is the ranking defined?
It's defined based on this gradient-based operation. So you're looking at the gradient for that class. Say class 1, you look at the partial derivative at each feature map for that class, and you're seeing, what are the activations.
So every iteration is done.
Yeah. Yeah. What do I do?
What do you actually do with the data?
No, then you use it to compute these maps and you stack them. It's not just a stacking, there is a nonlinear operation to aggregate them, but you're using those boxes as an input now to that layer.
But I saw the purple layer is your output.
That is the final output, yes.
So how does all these green boxes influence the
So do you see this orange arrow that goes back in there? So once we compute those green boxes, we non-linearly aggregate them back, and we feed them back into that operation in there. So all of this can happen in the same iteration loop. It's non-linear, but yes, there's another weighting relation happening in it. Yeah. Yep.
That's what sense you represent. The environment will say for giving you or class.
Yeah.
And another is an segmentation level that applies to the segmentation, but now you're looking to reduce the label, right?
Yeah.
So basically, and then you extract it together, you connected them in order to use some features, basically, not to run to another set, right?
Yes, it's just to make it end-to-end and to see if...
so you are reusing it and you are saving some convolution.
I'm not reusing in the sense that something is saved and then...
Some features are reused, but you're not generating from the image twice.
Oh, yeah, correct. Yes. Okay, cool. All right, so earlier you asked a very good question is how do we... Actually, before I get there, sorry, I want to talk about more of these shape priors. So I'm calling those shape priors, but in reality, it's kind of an abstract way to define that. But there exists some real shape priors that you can have for these images. And one of them is the actual containment and exclusion hierarchies between the different elements of these objects that you're trying to segment. So remember, we're segmenting these glands, but in reality, these glands are these multi-part objects that have a central area that is in purple here, that is called the lumen, and that area is always surrounded by a thick border, shown in yellow here, which is a border of epithelial cells. And all of that is generally within some stroma cells, like some stroma tissue. So this kind of topology is always preserved when you look at glands, and that's regardless of whether the tissue is malignant or benign. So you always have lumen inside of epithelium, and both of these excluded from stroma. So we thought, okay, now that we have that, how can we actually make the network aware of that type of relationship? So we thought we can add some form of regularization to the loss that would teach the network, or tell the network, not to predict topologically implausible, segmentations. So remember, you're still given an image trying to predict a segmentation for these glands, and then further trying to classify the image based on that. But let's just focus on the segmentation at this stage. We want to tell the network that is segmenting that when you're predicting a class label for a pixel, you should know that whether you're saying this is a lumen pixel or an epithelial pixel, lumen should always be included within an epithelial border, basically. So respect discontinuity and exclusion properties. And so to do that, we introduced a way to have a hierarchy between the labels that the network is predicting. In other words, we kind of defined for the network what is a topologically plausible labeling. And then we redefined the way we're computing the softmax loss based on that labeling. So here in this equation that you don't have to bother about, we're just explaining that there is a validity indicator that we're introducing that tells the network whether the hierarchy or whether the labeling that it's proposing is plausible or not. And within that, when we're computing the the probabilities for each assignment for a pixel, we're also looking at the joint probability now between whether a pixel is from the class lumen, epithelium, or stroma. And then by using that joint probability, you can compute, you can kind of integrate this hierarchy between the labels. But there is another prior that we have when we look at the plants. That prior is that most of the time, I'm going to show you again this image, most of the time plants are these smooth objects. They're vertipart objects, but they're smooth. So there shouldn't be too many irregularities in the segmentations you get. So an easy way to enforce smoothness is to look at the neighborhood of a prediction, right? So for instance, if your network that is making pixel level predictions has too many irregular, too many irregular plans, then you want to enforce it. You want to enforce it not to do that. You want to enforce it to have these smooth borders when it's predicting plans. And so to do that, we just simply added a smoothness curve here that, given a pixel, looks at the neighborhood of that pixel. And if within that neighborhood, the prediction for that pixel shares the same label than its neighbors, we enforce that to have, we enforce the difference between two neighboring pixels that share the same label to be small in the probability space, right? So for instance, if you look at my graph there, if pixel P and pixel Q share the same label in the ground truth, then we expect the output of the network, the output probabilities of the network to have a small difference. And if they do not share the same label, so XP and X2 have a different label, then we don't expect the probabilities of the network to be that close, right? The difference to be that close. So that's how we kind of infer a higher order term, because now it looks at the neighborhood of the pixel. Does that make sense? Did I go very fast again? I guess I did. Yes, I did. Okay, two.
It was your speech, right? Getting the knowledge of my ignorance.
Oh, I'm sorry about that. I can go slower if you feel.
No,
you're fine. All right. I tend to speak fast, so sorry for that. All right. But, so now that we looked at this, it's kind of the big idea of all this is that we're trying to create loss functions that capture the priors that we have about the objects we're trying to segment. And then that also capture more higher level prior than just pixel-based or image-based information. So back to your question earlier about the weighting of each coefficient of the loss. There is state-of-the-art out there that looks at how to weight the losses in terms of importance when you have multitask type of cases. So we look at something similar here, in the sense that we thought, okay, so we have this loss function now that has four terms. One is for the class of the image, one is for the segmentation part, and the two other terms are for the topology prior and the geometry prior on the segmentation. And we, most of the time, have a weighting coefficient in front of these terms, and the way you do it experimentally is that you would grid search over the parameters and try to find the right trade-off based on your validation set between these four terms. So what we looked at here is whether we could leverage uncertainty to identify that parameter, that hyperparameter in front of each term. And so in other words, you'd like to tell your network that if it's highly uncertain of a certain, for a given task, if it's highly uncertain of its predictions, then you'd like to penalize it more. And if it's less uncertain, then you just don't want to play too much with that term. And when we say uncertainty, it's very hard to quantify uncertainty when you're looking at deep learning networks, but one good way that was introduced in the state of the art is you could look at the activations themselves. So if your output activations are very uniform, then it means that your network is likely not making a strong decision towards anything, so it's highly uncertain. If your activations are more smooth or smoothly distributed, it's less likely that it's very uncertain, right? It means if it's highly non-uniform, it means that it's kind of the weights, the activations are kind of pulling towards one direction. So what you're saying is, okay, let's just train this hyperparameter, or that big of sound parameter, to based on that information, based on how uniform are the activations, just refine the coefficient in front of each cluster. And that's how we kind of got away with learning the right hyperparameter in front of each cluster. Yes? Yeah, so you see these two figures? Was this quite clear, or is this what you want to do? Okay, so here what we're looking at is kind of like an imaged version of the distributions of your activations, right? So that's also something I'm trying to say when these lands. It's not a very good representation, but you can think of the uniformity of your output this way. So let's say the predictions, the activations for my segmentation network are very smooth like here and very uniform. It means that it's not very much confident about each pixel's output, right? So in other words, when we use that information, we can now say if you're very not confident about that, you should probably penalize that specific term of the loss more than the rest, right? And that way is just introducing kind of a parameter in front of the activations f in that equation, because now you'll be weighting them. And with some derivation, you can get from that function, you can get to the loss function down there, where it's just a coefficient in front of each term. And I'm going to stop here because I'm timed out.
Thank you very much to Dr. BenTaieb, and we are very proud actually of having her here, and we're also very proud that Roche is one of our clients. So the next speaker is Dr. Michiel Schaap from Heartflow. Actually, also Dr. Schaap has been known already for a couple of years. They are also very supportive of BenTaieb, and this is where we met them for the first time. They are sponsors of BenTaieb like we are, and they also have been featured at least once in our magazine.
Thank you. Well, and great to be here. I want to talk about Heartflow, the Heartflow analysis, and how we use deep learning at Heartflow specifically. So it's going to be a combination of talking about coronary disease, the Heartflow analysis, and then I want to dive a little bit into why we use deep learning and how we do that. So coronary artery disease, as I assume a lot of people know, is a disease that affects the arteries that feed the heart with oxygenated blood. So in the vessel wall, plaque can build up and block the arteries, and thereby not providing oxygenated blood to the heart muscle. So about 1/3 of the global deaths in the world are because of coronary artery disease. And just in the US alone, 500,000 people a year die of a fatal heart attack. More than $200 billion a year is spent in the US alone on the diagnosis and treatment of patients with coronary artery disease. So it's a very large and impactful disease. And a big issue here is how do you find the right treatment for patients with coronary artery disease? So there are patients with symptoms, but what do you want to do with these patients, and how do you want to treat them as a doctor? So you can differentiate the disease, coronary artery disease, in two groups. One is non-obstructive disease, and the other is obstructive disease. So obstructive disease means that there's actually a plaque buildup somewhere that's blocking the transportation of oxygenated blood to the myocardium. And non-obstructive disease means the patient has chest pain, but it's not caused by obstructive disease. The symptoms are similar for these patients, but one you can treat, and the other you treat different. One you can treat with an intervention, so stent or bypass surgery, and the other you treat often with medication. So how do you find this difference? How do you know how you want to treat the patient? So what is the standard way of doing this is you have a battery of non-invasive tests. So all the patients with non-obstructive disease and obstructive disease go to these non-invasive tests, and you test for symptoms that are indicative of obstructive disease. If there is an indication of obstructive disease, you send the patient to a catheterization lab and you do invasive tests. So the invasive test could be coronary angiography, where you place a catheter in the femoral artery, you move it into the coronary arteries, and you actually measure the flow of oxygenated blood, or you take an image with higher precision to see the anatomy of the coronary arteries. The problem here is that with the existing non-invasive test, there are a lot of false positives. So a lot of patients are sent to the cath lab, the catheterization lab, without actually having obstructive disease. So a little quiz for the audience here. So positive predictive value, that means the fraction of the positives that you predict of the test that are actually positive. So what do you think is the positive predictive value of current non-invasive tests? So anyone that thinks that that is above 50%? No? I gave a little bit of a hint. So it is actually about a third of the patients that go to the cath lab actually have obstructive disease. So 2/3 of the patients that have this expensive and dangerous, risky test actually don't have obstructive disease. So that's something that we at Heartflow try to reduce, this number. Can we send less patients to the cath lab for an obstructive, for invasive tests to diagnose obstructive versus non-obstructive disease? The ethical side of
because some of those things are controversial.
Yes, indeed. There are people that make a living out of doing catheterizations, indeed, and that's something that we deal with, indeed. There is some pushback from the community. You're right. That is something that we are dealing with. Yes. Yeah, you're right. So indeed, if you look at the you look at the ROC curve of the non-invasive test, and you find a point where you reduce the false negatives as much as possible, and therefore you get a lot of false positives. Indeed, that's definitely true, and that's also why it's so hard to get an accurate test here, because you always have that part. So what do we do at Heartflow? So we have a non-invasive test that is actually more accurate than all the existing non-invasive tests. It starts with a standard coronary CT scan, and based on that coronary CT scan, we deliver a better diagnostic performance than the other non-invasive tests. And the idea is actually relatively simple. We try to reproduce an existing invasive test. So that's fractional flow reserve, also called FFR. And what FFR does, so it's one of the ground truths that are used in the cath lab. So if this patient is sent to the cath lab, this is one of the tests that is used. And it actually measures the blood pressure in the coronary arteries after a lesion blockage, and so distal and proximal, so before the lesion. And if this fraction, if there's more than a 20% drop, large clinical randomized controlled trials have shown that these patients need to be treated because there's a significant drop in the blood pressure caused by a lesion. And we do the same thing, but let me first show you why it's important to measure the blood pressure and not just look at the anatomy. So here you see a CT scan on the left, where you see a vessel. So here, this is where I'm now pointing with my mouse, is the aorta, and then there's a vessel flowing here. The white spots is calcium, so that's where plaque is built up and calcifies over the time. And there's actually a reduction in the size of the vessel, more than 70% in this case. And LAD stands for left anterior descending. It's the largest coronary artery that you have. In both patients, in case A and in case B, there was a 70% stenosis. But if you then look at the FFR results, on case A, this patient had only a 13% drop in the blood pressure, and in case B, there was a 29% drop in the blood pressure. So case B, according to these clinical trials, you need to place a stent in, and case A, you can treat with medical therapy and monitor over time. So what do we do at Heartflow? We take a CT scan. The CT scan is sent to us by the hospital to our systems in the cloud. We extract with deep learning an anatomical model of the coronary arteries. We compute a physiological model, meaning how much we simulate how much blood demand the patient has, where the blood consumption is needed, and then we use computational fluid dynamics technology to simulate and compute how blood is flowing through the coronary arteries of this specific patient. And thereby we compute the values of the FFR instead of having to measure it. And if you then compare us against the existing non-invasive technologies that are there, you see here, so talking about the ROC curve that we just talked about, is we have by far, we're outperforming all the existing non-invasive tests. And because of that, we can actually reduce the percentage of patients that go unnecessarily to the cath lab from 73% to 12%. And then of course you think about the false negatives. Are there patients that we send home that actually have events? And we're now a bit more than a year in large clinical trials, and we've seen that multiple clinical trials have shown that the patients that we defer from the cath lab are event-free, and actually the predictive value of patients with a Heartflow analysis value, we call it FFR-CT, FFR based on CT, of more than 0.8, are very indicative or it's safe to defer these patients away from the cath lab. Actually, 2 weeks ago, we demonstrated our first follow-up data of almost 5,000 patients that we followed over time. And there you see the difference in cardiovascular death and MI over patients with FFR-CT values of above 0.8 and below 0.8. And the values of above 0.8 are similar to what you see in the general population, and below 0.8 is actually similar to patients with obstructive disease. Yes, yeah, I'll talk about that. I'll answer your question in a... So, and also, which is interesting, actually, is we have a much higher sensitivity than existing non-invasive tests. If you think about the existing tests, like SPECT, which is a nuclear imaging modality, which has only a sensitivity of about 45–50%, and we are at 86%. We also catch more disease, actually, than the existing techniques. So you win on two sides. So to answer your question about the FDA and other regulatory clearances, at this point, we've been used. Actually, this number is now about 30,000 times to diagnose patients in clinical practice. We give results back in about 5 hours, which fits perfectly in the current workflow for diagnosis of patients in the cardiac pathway. The only thing that we want to work on is to be able to be used in emergency care. For that, we have to reduce to about 2 hours. We're reimbursed by Medicare and most insurance companies in the US and parts of Europe and in Japan. And we're cleared for use by the FDA, the UK, NHS, and most parties in Europe and Japan.
Why does it take 5 hours?
Why does it take 5 hours? A really good question. So that is something that I will answer in one of my next slides. So please remind me if I don't. So what I want to talk about, because this meetup is about deep learning and computer vision, how do we use machine learning? How do we use deep learning? And the answer to that question is that we have humans in the loop checking the results of the deep learning results. So if we want to talk about the image analysis, so we have the CT scan that comes in, and we want to extract the anatomical model and get the information to construct a physiological model. So you can think about physiological model are things like the size of the myocardium, for example, that we want to extract. So how do you extract this anatomical model of the coronary arteries in high precision? So why high precision? If you think about the blood flow simulation, so here you see two scenarios. On the left, it's the same patient, so the CT scan is the same, but there are two segmentations. So this projector doesn't really help, but there's actually an overlay in red here in the segmentation, and there's a small difference in the segmentation. And on the left, and you can trust me on that, it's a little bit tighter than on the right, but it's really hard to see the difference. And
Scenario 1 and scenario 2.
yeah, between scenario 1 and scenario 2, there's a small difference in the segmentations. But if you then look at the difference in the results on the blood flow simulation, so on the scenario 1, you got 0.68 FFRCT value, so 32% drop in the blood pressure, and on scenario 2, it was 91%, so 9% drop in blood pressure, with only a very small difference in the segmentation. So we need to be very, very precise in the segmentation of the blood vessels, and especially at the parts where the vessel is the narrowest in the MLDs. So MLDs means minimum lumen diameter, so the stenosis it's also called in medicine. So why is that hard? So here are some examples of how the image data looks. So it's fuzzy, it's unclear, there are artifacts. So the gray area is the blood vessel, where the blood flows, then the area, this dark gray area is plaque. It's where the plaque is building up in the vessel. The white spots is calcium. And we need to find exactly where the lumen is. And the lumen is the part where the blood actually is and can flow. But we have to deal, same as the previous speaker, with artifacts in the image data. So here you see a CT scan, and the white area is the blood vessel, but as you can all imagine, this is not the actual patient. The patient doesn't look like this. This was an artifact during the CT acquisition. That's because the heart is beating and the CT scan is made over multiple heart cycles. So the CT scanner tries to stitch these images together, and it sometimes fails, especially if the patient has irregular heartbeats or the discontinuity indeed. So this part, that's not how actually the blood vessel of that patient looked like. But we need to deal with that from the image segmentation side of things. We also have to be very robust to anomalies. So here's a patient with only a single coronary artery. So if you're from the field, this is very special. Like normally you have two on the left and on the right side. This patient had only one. Our algorithms need to deal with that. We also need to be able to deal with all sorts of types of diseases. So very large aneurysms on the coronaries, which in this case, this middle thing here is the aorta. This is a coronary artery, and it should be about the size like from there to there, but it's actually up to here, and it's larger than the aorta now, so meaning the vessel that's normally about 4 millimeters is about 40 millimeters or more there, and we have to be able to deal with that, and the same for the left ventricle. So how do you do that? How do you make an efficient, reproducible, accurate system that can interpret CT angiography? So when Heartflow was started, it's a spin-off of Stanford in 2007. There was no deep learning, computer vision, image analysis was not that far. The idea was we're just going to do this all with humans. With the current volume and the scale that we're looking at this year, we would need about 80,000 analysts that would do the analysis for us. And all the projections and the finances were based on that. Luckily, deep learning came around in 2015, and before that we already made quite some progress with other things like random forest and other machine learning techniques. So we use deep learning, but we combine it with quality control by humans. Because we are FDA approved, we need to guarantee that the results that we give back to our customers are accurate, and we're doing that guarantee by having a human in the loop that checks the results and guarantees they're accurate, which is great for me as on the deep learning side of things, because we get all the results back from this quality control. So the hospitals send us the CT data, our in-house staff of analysts review the data, and we store all these corrections on the deep learning results. So you get a huge amount of really valuable high accuracy annotations. To improve our system, yeah. So that's we get this cycle here. So here you see the whole pipeline. On the left, you have the physician that sends the data into our cloud system, cloud computing system. We do there the deep learning analysis. We get the model, we send it to our analysts, they review and correct, and it goes back into our system for the next iteration. Of course, then we need to go through FDA approval to make sure that that new system is also valid for like regulatory purposes. But then we can improve our system, and then we send the results back to our customer. So, can you just use deep learning out of the box to do accurate lumen segmentation of the coronary arteries, where you want to be very precise, subvoxel accurate, smooth, and it has to work, fit really well with the computational fluid dynamics. So if you take 2 years ago, this state of the art perhaps for 3D image segmentation, and you train this model on 4,000 images, you get about this. So you get good results, but they're blocky. There are gaps in the segmentation, sometimes there's spurious components, and we have, as the previous speaker also said, we have a lot of priors that we know about, we have a lot of information about the lumen and about the coronary arteries that we can inject to get actually better performance than this. So this is a high-level overview of our pipeline. And I'm happy to talk after this a little bit about the details if you're interested. But the idea is that we do the segmentation path by path, vessel by vessel, because in our pipeline we have a series of steps. We first find the large structures, the aorta, the myocardium, then we let that be reviewed by our analysts. From there we find the vessel paths, so where are the coronary arteries? We review that by our analysts, and then we have guaranteed topology, so we know where the coronary arteries are. Given that prior, we then segment the coronary arteries.
That goes to the smallest item?
Yeah, so what we do, coronary arteries, so the question was, it goes to the smallest item, so it goes to the size of the smallest coronary arteries that you can see in the we miss coronary arteries. Actually, I have a comparison against humans later in the slide, so you'll see nothing is perfect, but we're doing really well. And the idea is that we do the segmentation in this specific, given the center lines, so the vessel paths as we call it, we build a representation of cross-sectional images. So cross-sectionally to the vessel, we slice the 3D image data and we build a stack of cross-sectional images, and in that representation we do the segmentation. We use a little trick called a 3D cylindrical ring representation, which makes it for the insiders rotationally equivariant, and we use that in a deep learning system to predict a point cloud on the surface, so we don't predict voxels, where we say a voxel is inside or outside the segmentation. We actually predict continuously a cloud of points on the surface of the lumen. And those points, they can be between voxels to give us the subvoxel accuracy. And that point cloud is turned into a smooth surface that's blended together with all the vessels and gives us our 3D model. So this system is, at that point, this is about a year ago, we had 25,000 annotated cases, and this volume is growing rapidly. Here you see like the segmentation result qualitatively for from the system that we call Deep Lumen, so our deep learning-based segmentation system for Lumen. And what we did is we compared our performance to that of humans looking at CT. And of course then you need a ground truth. So the ground truth was invasive imaging. So we took invasive imaging data where we had catheters inserted into the patients and then measured the anatomy of the patient more precisely than you can do with CT. And we compared the anatomy with that. So on the x-axis in both of the plots you see coronary angiography, which is an invasive imaging modality and can be seen as the ground truth for measuring the lumen diameter. And on the y-axis you see the size of the stenosis that we extract from the CT data. We gave the same CT data to an expert as to Deep Lumen, and you see actually that given the same CT data as an input, Deep Lumen, the automated system, can get closer to the invasive imaging modality than an expert can. Also, so this is apples to apples. Here we compared to the same data. What we also did is we compared our numbers, so not apples to apples, not on the same data set, to published data that has been published in literature on the correlation between the assessment of coronary size in CT versus invasive imaging modalities. And there you see that we outperform any published data. And the interesting thing is that, oh, this is the wrong color, of course. I don't know why this changed. So, of course, Deep Lumen is automatic. So there are only two green bars here. The Bovers 2012 and Deep Lumen. Those are the two automated ones. I don't know what happened here, but all the others are blue, an expert radiologist. And what I find really interesting here from this data, and we triple check this, we get an accuracy of 0.21 millimeters with a voxel size, so a 3D pixel size of 0.4 millimeters in CT. So that means that we have about half voxel accuracy compared to a different imaging modality acquired at a different time in a different physiological state. So that to me was really amazing, that the patients, the state of the patient is so consistent that we can align these data sets so accurately and that you actually get the same size so close to the actual ground truth size. So then three things that we're working on for the future. One thing that in the Deep Lumen method we already had when we designed it, a building mechanism for extracting confidence scores. So the method is not just predicting this point cloud, but it's actually predicting for the insiders point distribution models, Gaussian mixture models. So it actually gives a measure of uncertainty of where the boundary is exactly, and that we can we're potentially going to use. So here you see a vessel on a stretched out vessel, and the color indicates the certainty of the Deep Lumen segmentation. We're exploring, and it's going to be a tricky question from a regulatory perspective, if we could potentially only have the regions be a QC where we have a low confidence of the algorithm, or that it's regions where we ask the analyst to put more focus on. The other thing that came out of the Deep Lumen model, although we didn't teach it that, and we didn't explicitly put in that it had to look for different types of plaque, the model was actually learning that. And it was actually pretty close to what human annotations were, without explicitly telling it that it had to look for plaque. So it can differentiate between soft plaque and high density plaque, and that's something that we're going to also move to in the future. And And obviously, as we talked about validating with invasive imaging modalities, we now train with CT data. We train with human annotations in CT that are potentially limited. So one thing that we're going to do is actually train our systems with invasive imaging data. So the input is going to be a CT data set, but our ground truth that we train against is going to be invasive imaging data, and potentially at that point we're going to learn patterns that humans cannot see and truly outperform human annotations in CT. And another thing that we're working on, and that's, so everything that I talked about now is improving our existing product and making the accuracy of that better. We're also going to launch a new product, so an extension of our current system. What you can do there is virtually change the anatomy of the patient. So you extract the anatomy of the patient and you can actually virtually open up the vessel and see what a certain geometry modification, what the impact of that would be on the patient. And we expect that doctors can use this to do treatment planning. It's often hard to determine like where do you place the stent. Yeah, it's a what-if technology indeed. It's you often have these situations where patients have multiple stenosis and it's hard to know which vessel to treat. And because we use simulation, you can also simulate the treatment potentially. So this is something that we're now doing a clinical trial on, and yeah, we aim to, it's all finished, the product. We hope to get this to regulatory pathway and launch in 2019, this year. So that was my talk. Thank you for your attention.
Thank you, Michiel. I could answer questions.
Elaborate on the annotations that you're getting back. Are they just segmentations or are they also scores for regression?
You mean the annotations that we get from our analysts? Yeah, so we store the corrections and the type of corrections that they make. So they have different tools to make the corrections and different ways to view the data. And we try to store everything that they did and like what they saw, and also try to figure out the motivation of the change. So we believe that that is really important for later when we want to potentially be able to explain certain segmentation results to physicians.
Thank you very much. Okay, now I have a question for you. We are thinking of preparing a new meetup in a couple of months. So the idea is this kind of initiative something that you like and you would like to come again? In principle, yes. Okay. So probably another one in a couple of months about medical imaging again. Thank you very much. So a couple of announcements. First, our CEO, Ron just confirmed me that in 2 months we have a new meetup about medical imaging, so be kind enough to have a look at the website of meetup.com and see when the final date. And second, I would like to thank you, Dr. Kumar, for explaining us how complex it is sometimes to have medical devices accepted in the surgery room and in the field. There is a whole change for the health professionals that must be taken into account. Thank you very much for coming. It was a great honor to have you. And if you are not already subscribed to Computer Vision News, please do subscribe. It's for free. You can subscribe to your friends and colleagues. Just go on our website and click on magazine. There is free subscription for everybody. Thank you very much for coming.